Thank you all for coming here today. I'm very glad to see you here at my dissertation. My thesis focus on the development of non-leaching antibacterial approaches on cellulose-based substrates and their antibacterial mechanisms. I'm very glad that Spirk has talking something about antibacterial resistance. Here I want to make some complementaries. Antibiotic resistance is the ability of bacteria to combat one or more antibiotics, and it makes more difficult to treat and requires alternative medications and even higher dosage of antibiotics. It is becoming an increasing threat to human beings. 33,000 people deaths every year in European country. The fail to address the problem will lead to 10 times more deaths by the year of 2050. It will become a major cause of deaths. It's more than that cancer can cause today. The burden of antibiotic resistance of spreading is equals to the influenza, tuberculosis, and HIV combined. 75% of infections are health care associated, which means that it can be minimized by adequate infection prevention. To solve these issues requires a wide cooperation and contributions from the whole society, including the government, the industries, the non-government agencies, and research institutes. So as we are doing research in fiber and polymer technology, and I am working this wood chemistry and pulp technology, we're trying to focus on develop the new methods for antibacterial functionalization on cellulose-based material using layer-by-layer modification. And also, I try to implemented the sustainability concept, the studies, and also dig into the mechanisms of antibacterial surfaces, which is make us to making a more optimum methods for antibacterial approaches, and also more sustainable. So why we use cellulose-based material, not only because we are working in this field, there is actually a huge demand of antibacterial products in the society. For example, the antibacterial tissues, the water filtration tubes, which can use the cellulose as a filling content, and it can also be used as a textile in, for example, hospital. Talking about the antibacterial approaches, there are two main categories. One is called leaching, which means the biocide can gradually leaching from the surface and kill the bacteria around. The biocide are including antibiotics, cytotoxic compounds, and also nanoparticles. The most popular one is silver nanoparticles and silver-based polymers. It becomes a problem right now is it has a concern that it will create some impact to the environment. For example, it constantly releases the compounds to the environment. And also, it will gain the risk of antibiotic resistance. Non-leaching, on the other hand, are using immobilization of the polymers onto the surface and killing the bacteria upon contact. So these components, including quaternized ammonium components, compounds, and guanidinium compounds, and also cationic polyelectrolytes. They can introduce the positive charge to the surface, which can be used to deactivate or kill the bacteria adsorbed. So the technique we use to create such a surface is called layer-by-layer modification. It has considerably attention recent years because it can making a nano-thin films onto the surface to realize different surface functionalizations. And it has an extensive choice of substrates, and you can actually absorb different layer materials by using one layer, either multi-layers, to realize different functionalizations. Our previous work we use cellulose fiber as a substrate, and making a 3-layer system out of PVAM, PAA, and PVAM. The PVAM is a cationic polyelectrolyte, has a higher positive charge on the polymer chain. PAA, polyacrylic acid, is a negative anionic polyelectrolyte. They can be assembled layer by layer on the surface of cellulose fiber. The whole process is water-based, and there's no toxic compounds are involved, and at room temperature. And the result is very interesting. It has over 99.99% of bacterial removal. So what is a bacterial removal? The bacterial removal is that by using the modified fibers and incubate with the bacterial solution for over 18 hours, and then examine how many bacteria left in the solution. And by counting the number of bacteria left in the solution, you can calculate back to know how much bacteria are removed from the solution. So the more bacteria left, the worse removal ability. The less bacteria left, the better bacterial removal activity. Besides the bacterial removal, we also check the bacterial growth inhibition. It's one of the important aspects to evaluate the antibacterial properties. So right after the bacterial removal test, we add 10% nutrient to the system. And after 18 hours incubation, we check how much bacteria left in the solution, and we remove the fibers. And we use optical density to see how clear is a solution is, and we compare that with plus control and references. So in current study, we know that PVM works good. So what about some other similar cationic polyelectrolytes, including polydetmac and PAH? Therefore, we make a 3-layer and 1-layer system on top of cellulose fiber, and then we evaluate the antibacterial activities. We found that actually it works pretty good. Most of them has over 99.9% of bacterial removal, and some of them can remove both gram-negative E. coli and gram-positive B. subtilis, and they reached near or over 99.99%. And also we checked the bacterial growth inhibition, and we found that the 3-layer PVM still stands at the top and has a very good results. It can inhibit the growth for both E. coli and B. Bacillus subtilis. Well, when we're talking about non-leaching contact active surface, the limitation is the surface area, because the surface only can work when bacteria are in contact. So the surface area is very important. Therefore, it's also important to know what is the capacity of the surface can absorb bacteria. So one gram fiber equals around 1 square meter of surface area. So if we assume that, let's say E. coli, if E. coli dense packed on the surface of fiber and form a monolayer, the theoretical maximum capacity will be 10 to 12 cell units per gram of fiber. And what we have in our 3-layer PVM system, modified fiber, in our previous standard test of bacterial removal, by calculation, we found there's 10 to 9 cell units. And then we think there's a lot of space that we can take advantage of this modified fiber. Therefore, we increase the bacterial solutions concentration and decrease the amount we're using of modified fiber. We reach the capacity of 3-layer modified PVM sample to 5.5 multiplied by 10 to 11 cell units, which is much closer to the maximum capacity. Which means if we use 3-layer PVM modified surface on the fiber, it has a great potential to have a longer lifespan and life cycle of the contact active surface. And what makes the difference of these different cationic polyelectrolytes? By using the nitrogen analysis, we can see there are much more adsorbed amount of PVM on 3-layer modified samples. But the absorption amount sometimes can strictly say it connect with the antibacterial properties, because if you have so much adsorbed amount, but your charge are all neutralized by the previous layer, then it should not work. Then it's very important to say to check what is the surface charge of these samples. Therefore, we use this polyelectrolyte titration, which can titrate the surface charge and determine what is accessible positive charge we have on the fiber. And we can find that 3-layer PVM also have a very high surface charge available for the bacteria removal. Since we know the 3-layer system works very well, what about we do a further step to try to replace the PEA by a bio-based cellulose nanofibril? Although CNF has less charge than the PAA, but it can actually create a surface structure and also generate the surface area, allows for the bacteria to contact. To evaluate this, I modified the pulp with first deposition 1 layer of PVM, and to find optimized adsorption condition, I use different pH setup and different salt concentration for CNF deposition. And then the third layer still keep the PVAM. By checking the adsorbed amount of PVAM, we found the CNF contained samples has a generally lower PVM adsorption. But within this CNF contained samples, the higher salt concentration at lower pH has a higher PVM adsorption, which can be expected as a better antibacterial behaviors here. So if you take a look at the bacterial removal, from 10 millimolar sodium chloride and CNF contained samples up to 100 millimolar, the bacterial removal ability is even better than the PAA contained samples. Also, the great result from this modified samples with CNF at the higher salt concentrations, it also has a much higher growth inhibition. So it's reasonable that between this CNF samples, the more PVM absorbed results in a better antibacterial activity. But interesting result is why the generally lower absorbed of PVM samples has better results than a relatively higher PVM absorbed samples. To study this, it is quite impossible to study on the scale of fiber because the surface of the fiber is very heterogeneous. And if you're using fluorescent microscopy, the fiber will autofluorescent. It will hinder the signal from bacteria, and it makes it impossible to visualize. Therefore, we need a smoother and more homogeneous surface. Therefore, we create these cellulose model surfaces based on by spin coating the dissolved pulp onto the silica wafer, and we modify this surface exactly the same way as we modify the fibers. And then we can see the difference between those. First, we can see that at lower pH, the samples the CNF tend to form these aggregates and island-shaped structures on the surface. And with increasing salt concentrations, we can see that the aggregates become bigger and it becomes larger. And then we seed the surface with bacteria and visualize under fluorescent microscopy, and we can see that more bacteria are focused on this island or aggregates regions rather than the flat regions. And if we measure the roughness of the islands region or aggregates versus the flat parts, we found they have a much higher roughness. So a plausible explanation about why the CNF samples are better than the PPA ones, we think is because we introduce a CNF to the system and give it more surface area and more roughness to allow the surface to capture and trap the bacteria. So far, we only considered about bacterial removal and bacterial inhibition. It's more about the absorption between the bacteria and surfaces. But we have no idea about what the bacteria feels. Are they alive or are they dead? To study that in a controlled way, we try to create surfaces with different charge, but exactly the same system. Therefore, we still borrow the concept using this cellulose model surface, and we coating the surface this time with different charge of the pulp, dissolved pulp, and then we modify those surfaces with the PVM, a CNF, and the PVAM. At this condition, the CNF works good, but also keeps less aggregates as possible. And then we get this positive charged surfaces with the increasing charge gradient. Since there's a lot of process in this surface preparation, we need to make sure that after the surface treatment, this charge gradients still kept the same. Therefore, we use the AFM colloidal probe, which is the way to measure the interaction force between the surface and the probe, and then using the LbL theory to fitting this force curve, we can get the surface potential. And using Poisson-Boltzmann equation, we can calculate the charge on the surface. So we can see after this process, the charge are still increasing from 7 to 47 millicoulomb per square meter. And then we seeded the bacteria on these different charged surfaces. First thing we can observe that with the increase of charge, more bacteria absorbed. And the way we're using to differentiate the live bacteria and dead bacteria is using these live-dead stains. There are two different stains. One is cyto9, it's a grain, which can penetrate all the cells. And the red one can only penetrate the compromised cell, which means, and replace the green dyes. Therefore, we can see that the red signal here shows the bacteria has a disrupted membrane, while green means the bacteria still has an integrated cell membrane. So we can see with the increase of the charge, the bacterial amount are increased, and also the dead ratio are increased from 4% to around 30%. Since we know the surface charge or surface potential, and we also can find the surface potential of the bacteria we use, then it is possible to actually calculate the force between bacteria and the surface using this Poisson-Boltzmann equation with the Gouy-Chapman model. The force was actually calculated, and we found that from the lower charge to higher charge, the pressure can range from 127 to 355 kilopascal. And if we assume that the contact area is around 0.1 micrometer, and we can get the area of contact around 1.5 multiplied by 10 to minus 13 square meter, and the force can be calculated around 50 nanonewton. But if this area sometimes are reasonably surpass this area, for example, if the bacteria are more elastic, then more contact area is, the F is even higher. Then we come to the question, is this force high enough to disrupt the bacteria? There are two hypotheses. One is if we consider about the Young's modulus of the E. coli, if you check them in different publications, it has a huge range from 0.05 to 221 megapascal. And our pressure is around 127 to 355 kilopascal. Well, if we assume that our bacteria can land it around here, then the force we have can actually disrupt all the bacteria on the surface. But the result is we only kill around 30%. And it's really hard to say if this is really the reason to disrupt that. But we can suggest that the modulus of our bacteria could be somewhere landed between this area. So that's caused like 30% of bacterial disruption. And there's also another theory is that if the surface has some structures, for example, our surface with the cellulose spin-coated and also with the CNF adsorbed, it has the roughness in the liquid range from 10 to 18 nanometer. It creates some tension within this bacterial membranes. Based on the studies before, the force to disrupt the cell membrane has been reported from 4 to 27 nanonewton. But this force depends on what kind of tip you are using. So it depends on the tip geometry. There is a correlation between the force required to disrupt the membrane and the tip geometry and also the indentation depths. This is called the Sneddon model. So it means that if our force is 50 nanonewton, and if we have, for example, a sharp enough surface or structures, it is possible to break the bacteria. But it's hard to decide which is the reason for our antibacterial surfaces. But one thing we are sure is that the surface structure really matters. So therefore, based on the study we have, we decide to design a surface with high positive charge and also with micro and nanostructures, which was believed to be lethal to the bacteria. So what is the function of charge? The function of charge is it can capture and trap the bacteria, it can generate force between the bacteria and surface, and it also can unstabilize the cell membrane. About this stabilization, there are some studies supporting it. One mechanism is called phosphate sponge model, means that the bacteria on the high positive charge surface, the lipid molecules will be teared apart and cause the unstabilization. There's another theory about ion exchange model, is that the native ions such like calcium or magnesium of the bacteria will also exchange with the high charge surface and causing the instability of the bacteria. The function of structures is basically physically rupture the bacterial membrane. For example, the cicada wings, it has this nano sharp spikes on the surface. It even don't need any extra force. It can tear the bacteria apart. So it is a very interesting thought to combine these two function together to create this synergy effect. Therefore, we create patterns and surface structure using femtosecond laser irradiation to create these micro and nano structures. And then we're doing layer by layer modification using our superior system, PVM with PAA and the PVM. The reason we didn't use CNF is that we don't want to disturb the surface structure, so we're trying to make surface as smooth as possible. So we got different surface topography, less spacing and larger spacing. The less spacing has more nano-shaped structure in it, more sharp or a little bit random. And the larger spacing, we have a more unified structure and patterns. And we also determined the surface potential using AFM colloidal probe. And we found that the unmodified surface and one-layer modified surface, there's a big difference between the surface potential of flat surface and pattern surface. But after three-layer deposition, it becomes more closer, which is good because we want to compare different structures at similar charge densities. So the result is very stunning. If you see here, the left part is a pattern area, the right side is a flat area. There are very clear border between the dead bacteria and the live bacteria. If you check the bacteria absorbed on the pattern area under SEM, you can see a lot of surface disruption of bacteria on the tips of the structures, which give us this hypothesis is like surface membrane was weakened by the charge at the beginning, make it more vulnerable to disrupt. And then when it comes closer to the sharp edge of the structures, the membrane can be ruptured. So if we put all the results here between two different bacteria, gram-negative and gram-positive, we can find out the surface works much better on gram-negative bacteria. And after 18 hours incubation, it has a huge difference between the pattern area and flat area. But Staphylococcus aureus, as a gram-positive bacteria, has much less death ratio. A plausible explanation about that is because the difference of the cell wall or cell envelope structure. First of all, the gram-negative has a thin peptidoglycan layer and outer membrane, which are a little bit softer. But for gram-positive bacteria, it has a very thick peptidoglycan layer, which are more stiff. On the other hand, the surface potential of E. coli and Staphylococcus, the surface potential of E. coli is larger than Staphylococcus aureus. Therefore, it makes sense that the E. coli are more vulnerable to our surfaces. After this bactericidal tests, we also examine the retained functions of the surfaces. The retained function of surface was done by using a new fabricated surface and used surface, which we put the surface into the bacterial solution and taken out, washed the bacteria and used again. Or used with bacteria means that we take the surface, incubated with the bacteria, and taken out and put back into the new batch of bacteria. Of course, we see this decreasing of retained functions, but they still keep pretty good functions if with the bacteria removed from the surface. Which means that the surface has a good potential to be reused. To check the compatibility between our surface modification and human cells, we do this cell culturing on the surface of modified surface. We can see that there are the surface actually support the cell growing, although it's not as much as this fibronectin coated surface, but it doesn't really do any harm to the surface to the cell, because the fibronectin coated pattern surface, they usually to promoting the cell adhesion and cell growing, and also make the cell thriving. So this test shows that the surface is not toxic to the human cell. So to wrap up this work, first we found the 3-layer PVM is outstanding and is the best antibacterial, shows the best antibacterial functions among other cationic polyelectrolytes. And by using the bio-based CNF at the middle layer instead of a PAA, it actually results a better antibacterial activity, which could due to the increased surface area and roughness. And we try to find out the correlation between the charge and bactericidal effect. They are positively correlated, and the interaction force between the bacteria and the surface can be actually calculated or estimated, and which is possible to disrupt the cell membrane. That is more depends on what is a surface looks like and what is surface geometry. And based on those research, we developed a new bactericidal approach to combine the surface charge and the surface structure. It has a very good antibactericidal effect, and it is reusable and safe to human cell. So after this, it's just the beginning. At least it's paved the way of future studies. For example, there are a lot of space that you can put new sustainable alternatives like lignin-based polymers, extract bark extractives, or chitin derivatives to replace the cationic polyelectrolytes and make a great antibacterial surfaces. And a further clarification of the antibacterial mechanisms are also required because we know the bacteria is not just a simple colloidal particle. It is a living organism. It has a pili, it has a flagella, and it also has a different functional proteins on the surface. It can interact with the surface, make the whole system more complicated. So we need more cooperation with certain expertise, microbiology, nanomechanics, and biomechanics. And also based on the studies, it's good to implement the sustainable idea for new product development in the area of a different industry, for example, medical device, hygiene product, or implants. And it's possible to working more closer to the industry and understand what they need. So at the end, I would like to give my huge appreciation to the founding Charles Scholarship Council of FOSC and my scholarship, and also my supervisor, Monica Ick, or a long time support, my co-supervisor, Dr. Josefin, Illergård, and the scientific advisor, Lars Wokban. And also I want to thank my cooperators, Dr. Torbjörn Patterson, Alessandro, and Professor Anna, Professor Frank, and Joran from Department of Micro and the Nanosystem Department. Yeah. So thanks for your attention.
Thank you very much, Chao. And now I invite the opponent to enter the stage.
Stop the live stream.